Several lectures ago, I made you a promise. I said we would do something with character matrix. We would talk about these things. And today we're going to deliver on that promise. Let's make this a little bigger. There we go. All right. So let's move that over. There. Perfect. So we can see our little brachiopod. Got to watch that bad boy spin. That looks good. All right. Character matrix. That's the term. And it's going to be a list of the different characters, exactly like you would use when making a species. Each of those body parts and the state of that body part, present, absent, long, short, but not just long, but like longer than 50% of the valve, like a definite rigid criterion that does not change with age meaningfully. Those are the things that you want. And so those are all going to be called characters. And so the question is, all right, when you're making a list of characters, once you have a list of characters, you can use these to actually reconstruct a family tree. Because we can do that with genetics. We have like A, C, G, and T nucleotides, and you can put it on a tree, and like sometimes this one changes, and so yeah, you can build your tree from that. You can also build a tree from these morphological characters. And so it's going to be median septum, present or absent, 1 or 0. Great. And then if 1, then is it long or short? And so that can be like 1 for long, 0 for short, and minus for it's absent. Like I already said, there's no median septum because it doesn't have a meaningful character state if the character itself is not present. Just like if you have a deletion in a protein, you can't imagine what the nucleotides would have been in that deletion because they're not there. You just need a way to deal with those that is reasonable. Yeah. And these are the most reasonable ways we have come up with in science to do these things. So, all right. So yeah, we can make phylogenetic analyses, figure out how things are related, even in the absence of molecular data like DNA. And this is how, with characters and character states. Useful characters, though. There's an art to coming up with these, and it's not that hard. And once you figure it out, you'll say, oh, okay, yeah, of course, that's stupid. Yeah. As with most things, once you understand it, it wasn't that big a deal. Getting to that point, that's sometimes the big deal. So anything that's distinguishing could be used as a character. Sure, thanks, Meyer, 1953. But this is a better description, so they're going to get better as we go down. So Pimentel and Riggins, 1987, a character is a feature of an organism, that can be evaluated as a variable with 2 or more mutually exclusive states, meaning either it's longer than half the shell or it's shorter than half the shell, and you have to include half the shell in there, so maybe shorter to half the shell or definitely longer than half the shell, whatever. You're going to define the characters based on natural groupings. You're going to look at a lot of these things. You're going to say, oh, usually it's only this long, but then occasionally it's this long. So if we just put hearts again, love it. If we just put our dividing line in the center, then we can separate those two things into biologically different, meaningful states of the character. Okay. A character is a theory. The 2 attributes which appear different in some way are nevertheless the same. Now that's kind of deep. What we're saying there is that there are probably similar genes and processes behind a character. So if a median septum can be long or short, we're assuming it's kind of the same genes that are making the median septum. They just stop here or they keep going all the way to here. It's like degree of control that is separating out these things. And so if 2 body parts appear to be from the same origin, and you can look at their development, you can look at their evolution in different taxa over time, then that's going to be a good clue that, these actually belong as different states of the same character. If there's no evidence for that, if you have early versions and you have a part in the same place, but it may have multiple origins, then it's best to keep those as separate characters and different states of those separate characters, even if it's just present or absent, the simplest state of them all. Yeah. So if the characters are homologous, we would expect that once you make the tree, they're going to end up clustered together and you'll have other branches that don't have that character, and these branches that do will all be branching from a common ancestor. Right here where my wiggling thumb is. Analogous characters are going to be characters where you have convergent evolution. Basically, you have a similar thing, like you have a shell that's wider than it is long. That could come from a lot of different types of shell. Like you can have mollusks that do that. You can have arthropods that do that. Yeah, it doesn't necessarily mean that these things are particularly closely related. So analogous characters are going to end up spread all over the tree. Homologous characters are going to end up clustered on the tree. And so you will assign all the character states blindly and as fairly as you can. Then you reconstruct it and you see, oh, yeah, these things group, these things don't. This is mathematically the simplest solution explaining the evolution of this group. And so that's what you go with. Yeah. All right. Other important thing about characters, they can be continuous or they can be discrete. Discrete is like yes, no, or longer than 50%, less than 50% of length. Yeah, something like that. Some criterion where you can say, yeah, for sure. Continuous characters are also useful. And these would be like the exact ratio of width to length, 1.02. And in another genus, it might be 1.04. Yeah. You can develop these based on lots of individuals. If there's a common pattern, you can develop it based just on the type specimen, which is probably a little less stable and a little noisier, but that's also a thing you can do. Yeah. So discrete characters, usually coded for presence or absence, occasionally type. Continuous characters are part of a range, and you just put the number in there because there are phylogenetic tree-making methods that can handle numbers in addition to discrete states, which would be like A, C, G, T on a DNA tree, or like, has a nose, doesn't have a nose on a character tree. All right, that was a lot. I would imagine you have questions if this is class, so ask them. Ask them, yeah. Okay. So, let's look at some characters. So these are characters distributed across the vertebrate animals with a spinal cord and bones. And so a shared character that's found in all of these things is going to be calcium phosphate bones, which everything that has bones has calcium phosphate bones. And they all share one common evolutionary origin, and it was preserved. It contributed to the survival of all of these things. There are no animals that have calcium carbonate instead of calcium phosphate bones. There are animals that under-mineralize their bones. Think like sharks. And we say, oh, they have cartilage bones. They have bones that are pretty bad at putting calcium phosphate on there. But genetically, they have some of the same equipment. It's just that some of the genes are maybe broken or don't work as well. And so mostly their bones end up a little bit bendier rather than mineralized calcium phosphate. But if they could mineralize, it'd be calcium phosphate. And their teeth are calcium phosphate, and that's how you can tell. Yeah, and those are made with the same genes. So a plesiomorphy is going to be something that's shared across the entire tree. Here, let's move this over a little bit. Yeah, there we go. And apomorphy is going to be something that is different. So here we're going to have tetrapods branching off. We're going to have everything with 4 legs down this branch of the tree. And we'll have things that do not have legs. Like the concept of legs has not yet been invented, and they're going to branch off over here. So things like nice little angelfish. And so having 4 separate limbs with bones in each limb is going to be an apomorphy that's going to define the tetrapods, which include all of these things, and you and me. Synapomorphy is going to be a name for a shared difference, so like a shared trait which sets them apart from the other things. So that's another, synapomorphy is shared. Apomorphies can be shared or not shared. An autapomorphy is something that is specific only to one of them. So the common ancestors do not have like a little bill. So the common ancestors, so having the bill on a platypus would be an autapomorphy. Having a pouch on a kangaroo would be an autapomorphy because it's not found higher up on the tree in any of the others. It just defines this group. Having a placental would define placental mammals like an elephant or like a person, and so we would be branching over here. And so if it's a character that only shows up in one branch, then it's an autapomorphy. And autapomorphies are actually not useful because you can't group anything by autapomorphies. They are just spread out at the tips of the tree. They are, about as useful as having different names on the branches of your tree. So what you want to do is you want to build up your tree in such a way that you don't have very many or maybe none of these autapomorphies. The synapomorphies are going to give you the branching structure, are going to reconstruct ancestors. It's not our differences that are useful in taxonomy. It is our similarities. It's the things we have in common. So yeah. Think about that. Yeah. And so, if you wanted to round out this tree a little bit, you can put in another marsupial, put in an opossum, which would also have a little rudimentary, it's not a very good pouch, but something like that. Put in a person, or put in a dog here, and then it would group with the elephant, and all of a sudden, that autapomorphy would turn into a synapomorphy. And that's what you want to do. Yeah. Plesiomorphy being a thing that is in the entire tree all the way across. Yeah. Ancestral means something that was probably present in all the ancestors because it's present in all or most of the terminal branches, like the things that are around now. Derived is something that changed, like it, some difference in evolution that led to a difference in outcome, a difference in phenotype. So you get derived traits down here near the tips, and you get your ancestral traits up here near the root of the tree. All right. And so what you can do is you can say, okay, criterion 1 could be, has calcium phosphate bones. Like, yep, check, we've got it. Criterion 2 could be, has 4 legs. And, 4 legs with limbs, 4 legs, but they're just jelly, something like that. You can have different character states of that one, and so on. And this is what a character matrix looks like. And this little list of 1s and 0s, and you can put question marks if you can't tell because it's not preserved in your fossil, and it could go either way. You can put a little minus sign if it is absent in your fossil, like if you have one character for median septum present or absent, and if it's absent, then it can't be long or short because it's not there. So you put a little dash character there, and the phylogenetic software will know what to do with that. Yeah. So you're going to code everything you can see and turn features into numbers in a table like this. And then we're going to read these through our tree making software exactly as if that was A, and that was C, and that was G, and maybe that's T. Yeah. And it's going to build us a family tree because 1 can change to 0, 1 can change to 2, 0 can change to 1. Yeah, it's just like mutations, only it's mutations of entire body parts that are going to be the things that make up the branches in our tree. Okay. So, this is pretty heavy on phylogenetics. Yeah, apologies for that. This isn't something you're super familiar with, but it should be something you've run into before. So, here we have a thing you can do. And so you can use computational statistical methods. And so if all these little tips, these are all the things that are alive now. This is like all the current generation. We can infer that there are shared characteristics, since there must have been a common ancestor, because there are a lot of shared characteristics in these 3 groups that are not found in the others. And so, bam. Do you remember our term for that? OTU, Operational Taxonomic Unit. Yeah, all the little dots all the way back on the tree are operational taxonomic units. Yeah, as are the tips as well. Basically, every dot on a phylogenetic tree can be. So OTU, OTU, OTU. And if every single child has a particular state, linear, like long and skinny, then you can say, well, if every single child has this, then probably the ancestor also had this state. And you can do it a little more sophisticated way, computationally, and you can go back and you can reconstruct even very ancient ancestors and say, in terms of probability, it's more likely that they had this versus this, based on the number of evolutionary steps it would take, and you always try and minimize the number of steps, because the simplest tree is the best explanation. That's our rule of thumb. Yeah. So, We're trying to keep it simple. Keep it simple. Yeah, all right. So we can reconstruct ancestral states. That's cool. Yeah. Defining characters can be challenging, just like it says. So look at these characters. This is from a paper, and it was talking about characters that are maybe not so good. These are characters that are maybe ambiguous. So, all right. Male anal point. So the way you set up a character is first you say what body part you're looking at. So sometimes, like if it's a brachiopod, you'll say ventral valve interior. Yeah, and so boom, that's the first part. And say, of that, which body part are we talking about? So in this case, we're talking about a male anal point. And like, okay. And then of that male anal point, what do we see? So we've got a reduced version, which means hard to see, and a distinct version, which means, I guess, easy to see. But where's the dividing line between there? So if you're going to make criteria like this, and these are probably not the best, you could probably do better, then what you have to do is you have to include a picture, and you have to show, like, here are some that are reduced, here are some other ones that are distinct. This is what I mean by reduced, this is what I mean by distinct. You have to either put it in words or put it into pictures, and it's often good to have both of those there, because your tree is of no particular importance, but others need to be able to reconstruct your tree and add on to it and add their other stuff there. That's the really important thing. And so it doesn't matter how you got whatever you got. It matters how well you can explain that and how other people can use it. Yeah, that's what makes something good. Yeah. This one, so ovate, like egg-shaped, or orbicular, like sort of spherical, I guess, maybe flattened. Is it that shape, or is it spatulate? And you'd have to go and look up what the heck spatulate is. I don't even know what that means. Stuff like that. So these characters would be less than optimal. You ought to be able to rewrite these characters and say, okay, male anal point, higher than 0.2% of the length of the shell. So that as the shell gets bigger, you still can say it's like really low, but you've got boundaries on it. You've got some kind of finite numerical boundary that says, yeah, you got to be this big. And then your character state 0, character state 1. Normally, also, we would say 0, which is going to be reduced, and then 1 distinct. So we're going to define our characters right there in the matrix, and that is going to be our little key that lets us put our little characters on a tree and let somebody else operate our character matrix and put their own things on there. Yeah. All right. So the most widely used format is that you're going to have first the location, so like where in the body. Second, the name of the characteristic or body part, typically, whatever it is that you are measuring. And then the state. And the states have to be mutually exclusive. It has to either be one or the other. There cannot be any in between. And you have to write the criteria in such a way that there is no possibility that it can be in between those two states. And you get to that point by examining a lot of different related organisms and figuring out that, it's always either this or that. And so if that's the case, then you can put your dividing point in between those two things, whatever it is. You just have to figure out how to express that in words, nice and tight, like a legal description. Like if you're making property, you have to identify the boundaries. Your yard goes to this point. You can put a fence right here. Yeah. And if you put it too far, it's coming down. Just like that. Yeah. So here are some coding strategies. So say you have some birds, and the birds can have a tail or no tail. If they have a tail, it can be a red tail or a blue tail. Great. You could break it up into different columns. You could just have absence of a tail or presence of a tail. So 0 is the absence, 1 is the presence. That would be a good way to do it. So 0s and 1s, nice. You could have column 2 be for color of the tail. And so you would call it tail color, basically. And if there is no tail, then you don't know what color it would have been. And so you put a question mark there because it's unknowable. Yeah. Or you can put a minus there because it's It's just absent. This is not a thing we should be considering. It's like deleted. And then in that case, you can have two states for red can be 0 and blue can be 1. Great. You can try to smush those all into one column. So if you think that there's a continuum from no tail all the way through red tail to blue tail, then you can put them all in one column. This may not be the best thing to do for this particular case, because if you think about it, it's probably different genes that are covering tail color versus absence or presence of tail. Like absence or presence, that's a developmental gene. That's like a Hox gene. Tail color, these are some pigments. This is some secondary metabolite. This is something the cells are doing long after they've either grown or not grown the body part. So in this case, I would not favor column 3. And then column 4 is showing a different approach. So you could define this as if a tail is present, like column 1, present or absent, then you could say presence of red tail and presence of blue tail. So this is presence of red tail, and so you got 2 1s, presence of blue tail, and there's the 2 1s at the bottom. That's a different way to define it, and there might be situations where you would want to do each one of these things. You've kind of got to look at it and you got to think, well, all right, if this is a creature and it is developing, and I know how similar creatures develop, are these controlled by the same process? Are there ever times when these two things are separate from each other? Or are these things always linked? Like if you're talking about hair color or eye color, is albinism a thing, albinism? Because if it is, then the hair color should be separate from anything else. It shouldn't be, presence of hair is brown, hair is, white or something like that. It should be, presence or absence of hair, and then separately, like, is this person suffering from albinism with very white hair, blonde hair, or is this person got, normally functioning color-producing pigment pathways, in which case they'd have brownish hair of some sort. There you go. All right. So you can compare these two shells. So these are actually very similar on the outside. Here, I'll dip into the presentation for just a sec. Oh, actually, no, we didn't separate it here. Okay, I'll dip back out of the presentation and put myself back over here. There we go. So these are two shells that look the same on the outside, but on the inside, they are quite different. And so you can look at these and say, all right, they're kind of similar, but look for the brachial ridge. Present in the bottom one, totally absent in the top one. And these are the same size from the same site laid down at the same time, which means that those differences are, those are real differences. Look at the median septum. It's barely there. It stops even before halfway through the shell versus it's pretty tall, pretty prominent. Yeah, that's a difference. You got this ring of large granules. Look at them sitting here on the edge, marking where the lophophore used to sit, versus no differentiation in size of granules. That's a difference. You've got these great big, thick andiridia right here, versus you've got very delicate, slender andiridia. These are the things like little fingers up here. That's a difference. So they're both andiridia. They're both probably serving the same function, but one is like really chunky and the other is very very light. And for that you would need pictures that go with it that show that, all right, here's what I'm calling a thick versus thin andiridium. Yeah, basically. Yeah, and you can do this for every body part and build up your characters, but you can separate out even outwardly similar organisms. And then once you have your traits, if you know when the fossils are from, you can actually use time to help separate these things. Because in order to be an ancestor and have an ancestral state, those would be the things that are earlier in time. And if those are all the fossils that you have, then your ancestor is somewhere within that group, probably. And so you can say, all right, what's the time range for each one of these things? And therefore, if you have the time range, you can say, oh, okay, then this trait must have come earlier. It must have been more primitive because it's found in the literally older fossils. Yeah. So time is your friend. And you can put this in. The fancy new way to do it is a thing called a fossilized birth-death model, which is the earliest appearance of the fossil and the latest appearance, and you use that to adjust and interpret the rest of the tree, which is based on the characters. And with a good fossilized birth-death model, you can actually put things that are living today and things that are fossils onto the same tree. And that's what everybody wants to do. That's like why fossils are interesting, because they show us how we got here. And even though they don't have DNA, we can still look at the morphology of things that are around now and in further morphology of things that were around then. And we can do it. It can be done. So, another prominent way to do this is with landmarks. And so landmarks would be particular points, and you're going to have a list of rules for this is how you define each point. It's going to be the point, the widest point of the cardinal process or whatever that is, farthest away from the midline, and then the one that's the most posterior on the midline, and the one that's the most anterior, but also on the midline. So that's your points 2, 1, and 3 in that order, and all these other points. And then you can measure what is the distance between these points. What's the curve between these points? What's the angle of that little arc between each point? And each of those can serve as a continuous data point. And you can use continuous characters just like discrete characters. It doesn't have to be 0 or 1. It could be 1.5 versus 1.6, just like that. Once you have all your discrete and continuous characters, you can do a thing called a principal component analysis, where you can basically organize all the traits. And so here we're organizing on one variable at a time. So we're organizing it, and then it looks like the main thing will be an axis that runs from small fish to big fish. So this is going to be length. And that's going to be the chief determinant. Most of the data sits along there. So that is the number one component of this. It's a statistical technique. The second component, which is going to determine how high this is off the line, that's going to be perpendicular. All these components are perpendicular, and you have to imagine multiple dimensions to make lots of components perpendicular, but don't worry about it. The math is easier than conceptualizing it. You don't have to conceptualize it in order to do the math. And so the second dimension is going to be height, which is mostly going to be determined by the overall length. But yeah, basically the differences in height from very tall fish of the same length to very short fish of the same length. That is going to determine, that's going to be the second most important variable here. And so principal component analysis, mathematical way of doing exactly that. You can do it with bird feathers. You can do it with points on the loaf of 4 of an extant brachiopod. You can do it with all kinds of stuff. Here is an analysis. And so we're separating these out. And these principal components, they can actually encompass multiple different characters. And so you can have a principal component that is like mostly length, but only a little bit of height. And so that's our first character right here. And you can have a principal component character that's mostly height, but only a little bit length. And that can be your second one, and so on for each of your characters. It'll be a little bit of each one of these characters being higher or lower than normal. And that together, that sort of fingerprint of coordinated change, will be your principal component. And so in these brachiopods look real similar to each other. But by measuring all those little points and calculating angles and lengths and distances between them, and then solving the principal components of these points, you can actually separate out these species. So here's 5 different species, and you can see on some of these principal component analyses, they're actually really different from each other. The difference between the principal component and the CV is a different style, canonical variate analysis, and they're saying that works a little better for this particular kind of brachiopod. That's not important. It's just a different way of doing more or less the same thing. So that was a lot, right? Where did we start? We started all the way back here with like principal characters. Oh my gosh, yeah, character differences. We went through trees. We went through how you code the characters into your little matrix. We went through some examples. We went for how you put time onto your matrix. We then ended up here with principal component analysis and actually showing the results. And so for each of these things, think of, I'd like you to kind of understand this. I'd like you to be able to do this. I'd like you to be able to interpret it. Like, what things do you need to make a good character? These characteristics. What things do you need when you're coding characters? Like, what's a sensible way to do it versus not sensible? If you can tie it to probable genetic programs of development, that's probably a good thing to do. And that probably takes a lot of book research into similar organisms. Yeah, or at least the most similar ones that still exist. So that's been a lot, I know. But it's important. And yeah, this is a complex field. I mean, we started out saying like red tail, blue tail, no tail. Yeah. And that's simple. That's kindergarten level stuff. But you can actually do some very sophisticated mathematics with this and with a bunch of linked characters. And you can end up unambiguously separating out species of things that look very similar to each other otherwise. So that's been plenty. I think on character matrices, I just wanted you to get a nice flavor for how it's done. And believe me, this well goes much deeper, but yeah, I don't think you need that for what you're trying to do. So thanks very much. This has been Character Matrices.